first of all, I'm really sorry that I'm not able to be there in person. I was at the airport this morning at 4 a.m. and my flight was suddenly canceled, and they told me the first flight to DC would be tomorrow in the evening, which would be very helpful for attending this meeting. So thank you for the flexibility in allowing me to present virtually, and thanks to the organizers for this opportunity to share our work, which I think is really exciting to be able to follow in Yingzi's footsteps. So I come at kind of the problem of fibrous dysplasia from a different perspective. We mainly focus on osteocyte biology in my lab at MGH. And we think of osteocytes as kind of conductors of the normal or beautiful orchestra of bone remodeling by making rank ligand, which activates osteoclasts, and making sclerostin, which regulates osteoblasts. Osteocytes have the ability to sense different cues to the skeleton, such as mechanical cues and hormonal cues, and then kind of relay that information to cells on bone surfaces. So we've really focused on parathyroid hormone signaling, knowing that osteocytes respond to PTH by turning up the expression of rank ligand and turning down the expression of sclerostin. And of course, this is the PTH receptor is a cyclic AMP-linked G-protein coupled receptor. However, kind of there are a number of dotted lines in these signaling diagrams for a number of years. So we kind of took it upon ourselves to try to map the signaling pathway downstream of cyclic AMP and PKA that leads to these changes in gene expression within osteocytes. So this led to the identification of kinases, which Yingzi already mentioned, called salt-inducible kinases. So these kinases are interesting because they're actually normally active in cells in the osteoblast lineage. And so as active kinases, they phosphorylate their substrates. And when 6 phosphorylate their substrate proteins, class 2 HDACs and CRTC proteins, this retains their substrates in the cytoplasm, where they don't do much. However, salt-inducible kinases are direct substrates of protein kinase A. And when PKA phosphorylates these kinases, it turns off the activity of SIK. And this leads to a reduction in the phosphorylation of SIK substrates. And so now these proteins can move into the nucleus where they regulate, where they serve as co-activators or regulators of CREB-driven rank ligand or MEF2-driven sclerostin expression. Kind of further complementing this model was the fact that because PTH signaling inhibits SIK activity, we observe that small molecule SIK inhibitors have kind of PTH-like effects. And really the connections to the potential connections to this pathway to fibrous dysplasia came as we started to analyze genetically modified mice where SIK alleles were missing in cells of the osteoblast lineage. So because cyclic AMP signaling inhibits SIK activity, we were kind of expecting a phenotype when SIK genes are deleted that are similar to what's seen with overactivation of the cyclic AMP pathway. And so we kind of started thinking about this in the setting of the Jansen's metaphyseal chondrodysplasia, which is due to an activating mutation of the PTH receptor. And when this mutation is put into osteoblasts and osteocytes, you kind of get this very high trabecular bone mass with prominent marrow stromal cells. And so we were very kind of excited and encouraged to see that when we delete SIK2 and SIK3 from cells of the osteoblast lineage with DMP1-CRE, we kind of recapitulate a similar phenotype. You see very dramatic increases in totally abnormal-looking trabecular bone with prominent accumulation of stromal cells in between that abnormal bone. These bone surfaces are full of trap-positive osteoclasts and, incorporate very high amounts of calcine and demeclocycline with dual labeling. At the molecular level, the changes in bone with SIK2/3 gene deletion are very similar to the changes in bone with the Jansen's metaphyseal chondrodysplasia allele. This is RNA sequencing experiments. So genes in the top right quadrant are upregulated by both SIK gene deletion and PTH receptor activation, and the same is true of genes in the lower left quadrant of the scatter plot. So there are many shared features between the phenotypes in bone seen with SIK2/3 gene deletion, constitutive activation upregulation of the PTH receptor, and of course, hyperactivation of cyclic AMP signaling due to these activating mutations in GS alpha, which are listed here. And at the molecular level, as Yingzi mentioned, we saw dramatic induction of genes in the Wnt beta-catenin pathway, as well as, of course, very high levels of rank ligand that's driving the differentiation of all these osteoclasts. So this, along with other data that I don't have time to summarize, has led us to develop this model, which is schematized here. But really, we focused in these publications on these actions in mature cells of the osteoblast lineage, as well as more recently in the proximal tubule, where some of the actions of PTH are through a Syk-dependent pathway. And so the major question kind of comes up is whether the signaling pathway might operate in kind of early cells of the osteoblast lineage before the commitment to differentiated osteoblasts, and could that have anything to do with the pathogenesis of fibrous dysplasia? So recently, we've kind of initiated projects to delete Syk alleles in different subsets of skeletal progenitor cells using a tamoxifen-dependent approach. And so we're using these 3 different Cre-ER strains, and along with tomato reporters, so we can follow the differentiation and behavior of the cells after the Syk genes are deleted or not. And what I'll show today are some preliminary data from a very exciting project that's performed in collaboration with Yingzi, as she mentioned, which is now funded by a DoD award. And so what happens when Syk alleles are deleted using Osteric's Cre-ER is shown here. So in this experiment, mice are given tamoxifen at 3 weeks and sacrificed 6 weeks later. And very similar to the animals where Syk alleles are deleted with DMP1 Cre, again, we get this dramatic accumulation of very abnormal looking trabecular bone with stromal cells in between. And when we stain with TRAP, an abundant accumulation of TRAP positive osteoclasts on these surfaces. And so now, because we have the TD tomato allele in this tamoxifen dependent lineage tracing model, we can ask if any of these FD-like effects are cell intrinsic. And the answer is it's complicated. And this might have been predicted based on what Yingzi mentioned about the cell autonomous and non-cell autonomous effects seen in her GS alpha activation models. So here are the cells marked with tamoxifen a couple weeks later in a control mouse with Osteric's Cre-ER and the TD tomato reporter. You can see there are a few cells that are present on bone surfaces. And obviously, we get a dramatic increase in the number of Osteric's lineage cells 2 weeks after TD tomato, after tamoxifen injection. In some experiments, we gave the mice EDU prior to sacrifice to see if the accumulation of TD tomato positive cells is due to increased proliferation. And the short answer is that it is. You can see that there's an increase in cells that are both positive for TD tomato and EDU in the when Syks are deleted compared to when Syk 2 and 3 are present. And then kind of interestingly, looking at whether these changes are kind of cell autonomous or non-cell autonomous, as we focus in on these accumulations of trabecular bone in the animals where Syk 2 and 3 are flocked with the TD tomato reporter, you can see that there are some of the stromal cells that have a stromal appearance based on their morphology, which are TD tomato negative, which is similar to what Yingzi mentioned in her GS alpha model. And then there are other cells in the lesions that are TD tomato positive. And so moving forward, we're very excited to examine kind of the interplay between these Syk mutant cells and their kind of neighbors, and trying to understand what factors might be secreted in a Syk-regulated manner that might control the ability of the mutant initiator cells to drive the expansion of these non-mutant stromal cells. So again, thank you so much for the chance to be here virtually and share our work. Hopefully, it can convinced you to some degree that inducible deletion of SIK2 and 3 in cells of the ostrich lineage leads to a phenotype that has some features that are consistent with FD, which are listed here and we've reviewed. And in the future, we're very excited to try to understand the relative contribution of SIK inhibition to the overall phenotype seen in FD, to try to understand the substrates of salt-inducible kinases that are regularly responsible for this phenotype, and to think about whether we might be able to pharmacologically activate SIK in order to potentially reverse some aspects of FD disease progression. And so I wanted to acknowledge the members of my laboratory, in particular, Faye Papiano, who's shown here, who's an amazing endocrine fellow, who's been very involved in this project. Hank Rodenberg has been involved in these collaborations, as well as Yingzi Yang. And obviously, we're very excited to receive this funding from the DoD to really expand and amplify these efforts. Thank you so much for the invitation to present, and I'll look forward, hopefully, to attending in person next year.
Thank you, Marc.